Cloud. All right, so we're recording, just so you know. I'm going to share my screen. All right, so here's where we were last time. We were talking about the kingdoms of the eukaryotes. So this is the domain eukarya. So remember that this was a huge jump in evolutionary terms. We start, life on Earth started actually not, so Earth, as you know, it sort of coalesced out of a lot of, matter crashing into each other. So the Earth was this big fireball for a long time. It was, essentially molten on the surface. But eventually things calmed down and we got liquid water. And so bacteria actually rose relatively quickly after that. Actually, sorry, I have a notice I have to get it. So let me a little side note before we get back into life and all that big exciting story. Before we start class, I wanted to give you a written message here. I want to give you everyone a heads up. This is a message from a student. There's a student in this class who has stress-related involuntary movements and sounds similar to the neurological disorder condition Tourette's. Both they and myself thought that it would be proper etiquette to inform you of this occurrence so that if and when it occurs, you all have somewhat of a forewarning. Please ignore whatever you may hear and continue as usual. If the tics become frequent and are disruptive, the student will remove himself so as to not disturb your learning. Thank you. Okay. And yeah, that's another reason I might, so be recording, that sort of thing. Okay. Any questions on that? All right. So as I said, so bacterial life actually arose fairly quickly after, things sort of calmed down on Earth. But then there was a very long pause in which all we had was bacteria and archaea, which I described earlier. Both of those are single-celled forms of life, single-celled organisms. They're living, but they do not have nuclei. They do not have things called organelles within their cells. Everything is essentially in one container, which is the plasma membrane that surrounds the cell. Now, eventually what you have though, and this only started to rise more recently in evolutionary terms, are the eukaryotes. So these are cells that all have nuclei, okay? So they're much bigger. They're literally like, in terms of volume, roughly 10,000 times bigger than a bacterial cell. So they're absolutely enormous and very sophisticated. They have a tremendous number of genes. So the eukaryotes can be divided into different groups. There's 4 different groups which are called kingdoms, right? The first one is protista. So what is a protist? Well, there's an example here you can see here of a protist, which is this is a paramecium. It's a single-celled protozoan that you find in just about any pond in the world. It has a nucleus. It has mitochondria, things like that. They're very sophisticated. They have, I don't know if you can see this, it's a little hard at the resolution on the screen, but it's ciliated. So these things, as I mentioned these last time, they look a little bit like a slipper shape and they have these little cilia, these little hair-like projections, and they actually swim around and they hunt for food, right? In ponds, they're incredibly complex biochemically. They have more genes than humans do, okay? So these are not unsophisticated creatures, right, living in ponds. And I mentioned some other ones last time, like the amoeba that lives in ponds also. Okay, so that's what's in protista. Protista also has things, interesting things like slime molds. What else? Certain types of algae, things like that, like the sort of pond scum you'd see in a typical pond. Those would all be within the kingdom protista. So there's at the level of the cells, their cells are very similar to ours. They have a nucleus and they have organelles. They're very complex. Kingdom fungi, also pronounced fungi or even fungi. All those are good pronunciations of this. This includes things like the mushrooms. So these are multicellular. It also can include single-celled organisms such as yeast. We use yeast for baking bread. Ringworms, things like that. Those are all within the kingdom fungi. Okay, mold would also be there. Okay. So that's a very important group because obviously, what a lot of fungi involved in is things like decomposition, right, of organic material. As you've probably seen out in any nature walk. If you see a rotting log, there'll be mushrooms and things like that growing there. Okay, next up we have kingdom Plantae, right? The plants. These are very sophisticated multicellular organisms, so they have a lot of similarities to us. We're of course kingdom Animalia, but the Plantae are photosynthetic. That's sort of their big thing is that so this includes certain algae, mosses, ferns, conifers, which are essentially evergreen trees, flowering plants. Flowering plants actually came relatively late evolutionarily. What else do we have? These are multicellular, usually with specialized tissues. Their cells are incredibly complex. They're essentially able to generate sugar such as glucose, and then turn that into things like starch, right? So if you eat potatoes or corn, there's a lot of starch in there, right? So that's something we're going to talk about later, starch. And that's very helpful for us because of course we eat plants. And then finally we have the kingdom Animalia. We belong to kingdom Animalia. Now this includes things like sponges. So do people know what a sponge is? Like a sponge underwater? Go ahead.
Like coral.
Yeah, it's sort of similar to that, right? Similar to coral, right? But so they live underwater. They're stationary, so they look sort of plant-like. If you were swimming around in Bermuda or something, and you'd see these sponges, and you say, well, it looks like a plant. But actually, they're animals. And we know they're animals because at the microscopic level, they actually have a lot of similarities to us. Now, sponges are kind of very primitive animals, right? They're just on the cusp there, but they are animals. There's worms, such as an earthworm, that would be an animal. Insects, right? Those are all worms, things like crickets and so forth. Fishes, those are of course animals. Frogs, turtles, birds, mammals, right? So we're of course mammals, and we'll talk about that more later. So all animals are multicellular. They have specialized tissues. We're going to talk about tissue more later. They're going to contain lots of complex cells. And animals are consumers. We ingest food. What kind of food do we ingest? Well, we ingest plants. And then we also ingest animals. Like for us, we're omnivores, meaning that we eat plants, but we also eat animals. So humans evolved to eat both animals as well as plants. All right, so let's do a question. Okay, so actually, let's jump out of this. So I have, I don't know if you guys saw the email because I sent it out kind of late yesterday, but we're going to use Top Hat, and it's 100% voluntary. Let's see if I can get to the Top Hat, please. So there's a code here for joining. So if you feel like doing it, you can. You don't have to, but it's just sort of fun. So I sent out instructions how to join. Basically, you go to tophat.com and you can set yourself up with, and you can't do it on a phone. The initial setting up of the account has to be done on a computer because you need to go to a website. Or at least I think it's difficult. It would probably be difficult to do it on a phone. I'm not sure if you can.
As long as you type in the entire URL.
Oh, you can. Okay, apparently you can do it on a phone if you type the URL. Okay, so you could try. Again, no pressure. It's not for points. It's just for. I hesitate to use the word fun because people say, well, that's not fun at all. But okay, fun in relative terms is being able to answer questions in class and get some little feedback. So basically what you need is you go to tophat.com, it'll have instructions. You set up an account, you put in like your Hilbert name. Do not put any payment information. We're doing the free account. We're not paying anything, right? So if they ask you for any sort of credit card, do not put anything there. And here's how you join. There's a join code for this class, which is 307988. Okay, so that'll get you in there. So again, if you didn't set it up, don't worry about it. It's really not a big deal. Okay. So let's see how this is going to work. I'll put that code on the board. 307988. 7988. Okay, so that's our course code. Let me see how this works. So this is all a little bit experimental. How do I start? Tell it's been a while here. So how do I go here? Present. Start presentation. Okay, we already did this question, right? But let's take a look. So let's just do this as a. We did this last time, but just this practice. Which of these is not a property of all living organisms? So again, you don't actually have to have this. You can just answer mentally. Okay, so we did this last time. Let's see how people do. If there's any recall, I'm just going to give 30 seconds. So I'm going to stop this in a second here. So I'm going to go ahead and close this out now. So I think if I hit responses, we'll see what's going on. Stop. Okay, so we got 11 people answering out of 14, and they said parental care of offspring. That's correct, of course. Okay. Yeah, so that's correct. Let's go to the next one. Oh, we didn't do this yet. Okay, I'm gonna stop. Hold on. Well, let's just, how do I jump out of this? We'll come back to that in a second. I think that's a little bit of kind of where we are. Did I talk about this? I did talk about it?
Yeah.
Oh, okay. So let's go ahead and do it then. Sorry. All right. So which of the following on the list represents the lowest level of biological organization? Again, typically they give you a minute. I'm going to go a little faster than I'll just be 30 seconds. And you can talk. When we do these questions, you can go ahead and talk to your neighbors. Like, feel free to talk and discuss. Okay. I say that and then people are completely silent. All right, here we go. I think that's enough time, right? So we're going to stop that. We're going to look at the responses. So the cell is, and that's going to be the correct one. Okay, very good. Okay. So it's a little fun because you get to see the difference between this and me just asking the question without the software is you often get a really good spread. Sometimes we'll get like, there'll be 4 answers and everyone will be evenly spread among the 4 things or something like that. Or maybe there's 2 answers that people really like. And that really helps us with, it helps me because I can see, where the confusion is, hopefully, and we then have a little bit of a classroom discussion. Okay. All right. Oh, there's another one. Which of the following includes prokaryotic organisms? Okay, let's do that one. Let's look at it. Third question. There we go. Also, it's early in the morning, so it's good to work your brain a little, right? That's the thing. Again, go ahead and talk to your neighbors if you want during these questions. It's perfectly fine. Okay, again, because of time, I'm just going to go ahead and get your answers in. People are debating. Get your just throw an answer in there. Sometimes it's better just to guess. Do a wild guess. All right, we're going to stop there. See what the responses are. Okay, so we got. Okay. Which of the following includes prokaryotic organisms? And we've got a really good split. So this is actually the nice thing about the software is we've got a good split between protists and archaea. Okay. Now those are actually both kind of on the surface seem like good answers, but there's actually one correct answer and one incorrect answer here. So I think people clearly didn't pick fungi or plants because those are multicellular, right? Now, if you look at protists, those are single-celled organisms like Paramecia, amoeba, things like that, found in ponds. And archaea are these sort of strange bacteria-like organisms that are like bacteria, but we know they're genetically different. Those are single-celled also. But one of these is prokaryotic, the other is not. So it turns out that the archaea are prokaryotes, meaning they do not have a nucleus, right? Pro means before, karyote actually means kernel, but when we talk about kernel, we're talking about the nucleus, right? So they're before nucleus. So archaea are similar to bacteria in the sense they have no nucleus. Protists, I just mentioned a second ago, they're actually incredibly sophisticated. Like if you look at a paramecium, you might not be impressed, right? Because you're like, you're a human and look at this poor little paramecium, right? It's only a single celled organism and you're multicellular. It actually has more genes than you do. It's very sophisticated. It has mitochondria, which are organelles for generating energy. It has a nucleus, so it has a lot of the same things your cells do. So that pond scum is actually way more sophisticated than you might think, okay? All right, so the correct answer then, of course, is archaea. All right, so let's go on here. All right, let's talk about, let's go back here. All right, categories of classification. Just making sure I didn't skip everything. All right, we've got domain eukarya, or the eukaryotes. Those are cells with nuclei. Don't worry about the supergroups. Nowadays, we actually break the eukaryotes up into, I believe it's 6 different supergroups. You're not responsible for that, okay? That's kind of a new thing, and maybe in the future we'll talk about that more than we will the kingdoms. But for now, we're going to stick with the classic thing. We're going to talk about kingdoms. We have kingdom Animalia. They're multicellular, motile, ingest food. We're looking at classification of humans. So next, remember the mnemonic deer King Philip came, what was it? Came over for good soup, right? So we have kingdom. King Philip is phylum, right? That's the next thing. There's quite a large number of phyla within the Animalia. We happen to belong within the Chordata. We have developmentally a dorsal supporting rod that stiffens our body initially when you're an embryo. That eventually turns into your vertebral column, and you have a nerve cord that's actually dorsal. Okay. So that's actually a very important distinction in humans and in phylum Chordata compared to other phyla. Most animals have their nerve cord ventrally. Like if you look at a lobster or something like that, the nerve cord is ventral. If you look at an earthworm, the nerve cord is ventral. We have ours dorsal. And so we think we actually came from a kind of worm, and there's some genetic evidence and so forth that indicates that we probably flipped over. Like one day your great-great-great-great-grandmother was a worm and decided to flip over and live life upside down. And that's why all animals within phylum Chordata have a dorsal nerve cord, which is very unusual among all the other, compared to all the other animals in the world. Now we're class Mammalia, which means that we have hair, right? That's one of our distinguishing features. We also have mammary glands for nourishing, the next generation. We're of the order primates, which are generally adapted to climb in trees. Family hominidae, it's hard to say, hominidae, it's hard to say early in the morning, adapted to walk erect. But that's not actually, this is, I took this from the book. It's not, obviously we're adapted to walk erect, but this also includes all the great apes. So what are the great apes? Gorillas, orangutans, chimpanzees, bonobos. Those are all great apes. Humans are great apes, right? So the term great apes is synonymous with the family hominidae. Now, this also includes the Neanderthals. It includes things like Australopithecus afarensis, all our ancestors, right? They're now extinct, right? Okay, so that's that family. Now we go to genus. It's Homo. What's special about Homo? Well, this includes things like Homo sapiens and also the extinct species Homo erectus, who were very similar to us. That was one of our ancestors. We have large brains, so did Homo erectus. Tool use was something that Homo erectus did. Homo habilis is another one. So there's a bunch of now extinct ones. Oh, Homo neanderthalensis is another one, right? So I put that in here. Those are all in this genus Homo, okay? So they're all extinct except for Homo sapiens. Now, That's a quick quiz here, like question here. Homo neanderthalensis, did they completely disappear or are they still sort of around in some sense? What do you guys think? Homo neanderthalensis, did they just completely disappear from the face of the earth? No, they didn't. Where are they? I'm looking around this room and I'm seeing there's a bunch of Homo neanderthalensis genes, and anyone who's of, I believe, European or Indo-European, that whole group, which is a huge portion of the world, like half the world, they all have Neanderthal genes in them. So what happened was there was Neanderthal boy and Homo sapien girl, or the other way around. There's, things happen, right? They got together, and we have a lot of Neanderthal genes in the human population now. Okay, so they did not disappear. We actually have that around. There's a bunch of their genes right in this room right here. Now we finally get to Homo sapiens. So again, when you talk about a species, you always give the genus name as well as the species. It's always this two-part name. It's called a binomial nomenclature. So we have, obviously, this includes modern humans, right? We're Homo sapiens. Who else is included in that? Well, the Cro-Magnons. Have you heard of them? They were like an earlier version of modern man. They were extremely adept at things like tool use. They had culture, they had fire, they had lots of things that modern humans have, okay? So that's all included within Homo sapiens, okay? Any questions about that, about where we lie? Okay, all right. We'll come back and we'll be talking more about that in the future at some point. Actually, we come back to that more in the second half of this course in the spring. Okay, so taxonomists assign each species a two-part scientific name, which again, bi means two. So and nomial actually has to do with naming things. So each thing has a two-part name. So this is called a scientific name. It's a genus name and species name. The genus name is capitalized, and both words are typically italicized. So Homo sapiens is humans. Pisum sativum is a garden pig. Oh, and who is responsible with it? I put a picture there of Carl Linnaeus, who came up with this system. The word science is derived from the Latin verb meaning to know. So science is a way of knowing based on inquiry. So biology is a scientific study of life. The way scientific knowledge is acquired is called the scientific method. Biologists and all scientists generally test hypotheses using the scientific method. So this may not sound very surprising to you, but it was fairly radical. And the first person who's really in some way since been credited as being the first sort of modern scientist was Galileo Galilei. Have you heard of him? He's an Italian, he was supported, I believe, by the Medici family. In his time, he was not a suffering scientist at all. He was actually very well known and very rich and so forth. But he was one of the first people that actually did experiments, and he would know, carefully the results of the experiments, and then based on those experiments, he would come up with hypotheses, and then he'd test the hypotheses, and then he'd further refine his ideas. So this was actually kind of a radical idea because in the past, what had happened was there were great philosophers. Aristotle was probably one of the greatest ones of the whole world. And they would come up with ideas and they'd write them down, and they sounded like very reasonable ideas. And for thousands of years, people would just simply quote Aristotle over and over again. And Aristotle basically wrote about everything in the natural world. And so why would you ever do an experiment if the knowledge was right there in the books, right? Well, Galileo Galilei said, no, let's actually try and do experiments to see how the world really works. And that was sort of a scientific revolution, and it's all based on this method that he pioneered. So how do you do this? All of you are actually scientists, and you probably didn't know it. You've probably worked with remote controls before, right? And so let's take an example of you dealing with a remote control. So you pick up the remote control, it doesn't work, right? You probably had this experience at some point, right? Or it could be some other, if you haven't used a remote control, you've used some other device, right, that doesn't work. Question, what's wrong? Okay, so you start with a question. So you're an inquiring person, just like a scientist. You make a hypothesis based on your observation. A hypothesis is usually a fairly straightforward statement that's like this: the batteries are dead, right? It's just a positive statement meaning It's just like, this is what's going on. The batteries are dead. Okay, so based on that hypothesis, you then make what's called a prediction. So the big difference, and a lot of students get very confused about this, is there's actually quite a big distinction between a prediction and a hypothesis. So a hypothesis is a general statement. It's usually very simple. The batteries are dead. A prediction is much more specific. And a prediction helps you to determine what's going to, do an experiment. So a prediction almost always has the form of an if-then statement. So you say, so I'm going to make, if I put new batteries in the remote control, then it will work, right? So that's an if-then statement, okay? And that's actually referred, there's actually a fancy term for that. It's called deductive reasoning. If I jump off a cliff, I will die, right? That's an if-then statement. It's actually a form of deductive reasoning, right? This early part here, when you take observations and you sort of collect them together and make a generalization, you've worked with remotes before, right? In your life, or electronic devices, often batteries go dead, right? Whether it's a toy or something like that. You take all that information that you've gathered over your life or through some observations, that's inductive reasoning, okay? So you've actually been doing these really fancy things. You've been doing inductive reasoning your whole life. You've also been doing deductive reasoning your whole life. You just probably didn't know and put terms to it before, right? Okay, so next step is, you've now done the inductive reasoning, the deductive reasoning. Now the next step is to do an experiment. So you get some new batteries or some batteries out of a box, and you say, okay, put them in there, and you see if the remote control works. So if the remote control works, then the data, the results, support your hypothesis. So it supports the idea, right? Now, here's where it gets a little complicated. So people say, well, I proved it. I proved my hypothesis to be true. No, you didn't, okay? And this is where students get really confused. Most scientists and doctors will never say they ever prove a hypothesis to be true, okay? They will never say that. If they say that, it's a little suspicious because that's not the way scientists and doctors talk. They're going to say the data or the results support the hypothesis. Because what's another possibility? Maybe you took out the old batteries, you put some new batteries in, but in doing that, you sort of shook this thing around, right? Maybe there was some loose wires or something like that, right? So maybe there was really nothing wrong with the batteries. Maybe it just had to be shaken or something like that, or something else was going on. So it's actually there's no real logical way to prove 100% your hypothesis was true. So all you say as a responsible scientist is say, look, this supports my initial hypothesis, right, and my prediction. Now, on the other hand, what if I put new batteries in, and I tested the batteries, I know they're new batteries, they're good. I put them in there, I make sure everything's connected up right, close it up, and the remote control still does not work. Well, in that case, what you can say is I can reject my hypothesis, and the technical term is you say I falsified the hypothesis. That you can do with 100% certainty or near 100% certainty. You can say, look, I put in new batteries, it still doesn't work. Therefore, my initial hypothesis, which was that the batteries are dead, was not true. Okay, so you can prove the negative, but you can't prove the positive thing, right? All right, so that's a little confusing for students, but we'll see more examples probably of this as we go through this. So here's the general steps we want to use for the scientific method. We're going to observe and generalize. That's inductive reasoning. We're going to formulate a hypothesis. We're going to then make a testable prediction, which is much more specific than the hypothesis and leads us directly to the experiment. Predictions almost always in the form of an if-then statement, which is deductive reasoning. We then do an experiment, or occasionally you may not be able to do an experiment, but you can do some more observations. And then basically you can falsify the hypothesis essentially with 100% reliability, or your data may support the hypothesis but never prove it. But you may you may then do more experiments, okay? Probably not in this case, because it's a very simple one, but if you're a scientist doing something very complicated, you might start to generate even more hypotheses and do even more experiments. Okay, so basically this is a very complex diagram, but it's basically going through what I just talked about. Don't worry too much about it. It's basically you have observation, you come up with a hypothesis. Here they're showing multiple hypotheses, which is a possibility. But for each hypothesis, you have a prediction, you would do an experiment, and you might reject the hypothesis. And they're just saying that each of these hypotheses are different possibilities. And eventually you get close. Essentially, the idea is you're going to get closer and closer to the truth eventually, right? The more experiments and observations you do, and the more testing you do. So don't worry too much about how complex this particular diagram is, but you get the general idea. The idea is that science is what's called an iterative process, meaning you keep sort of redoing it over and over again, getting closer and closer to hopefully getting a better understanding of how things actually work. So here's the things I've already talked about, but it's just here. The observation is basically you're observing and studying something in the natural world. Scientists use their senses to make observation. They also, of course, augment their senses with things like microscopes and all sorts of fancy equipment. Based on their observation, the scientist states a hypothesis and a tentative explanation for a natural event. So a hypothesis this is a very good definition here, a tentative explanation for a natural event. So this thing about observation is kind of really cool. You've probably heard of Darwin, right? You know who Charles Darwin is? So he had a collection of barnacles. Barnacles are very interesting from an evolutionary point of view. So he would study barnacles with microscopes and stuff. So his kids he had a whole bunch of kids, and his kids were just assumed that, well, all fathers probably did something similar. So they go to their friend's house and like, well, where's your dad's barnacle collection? But Charles Darwin was a consummate, he was constantly doing observation, either personally or he was sending letters to people all around the world, asking them what they were observing about the natural world. Very curious person. Okay, so forming a hypothesis. So this initial part, remember I talked about you have start with some general observations and then you form a hypothesis. That's inductive reasoning. This occurs when you use creative thinking to combine isolated facts into some sort of cohesive whole. So in a sense, you're kind of generalizing, right? Do that. Here's an example. Fruits and vegetables contain a lot of vitamin C. You're probably aware of that. People with diets rich in fruits and vegetables are generally healthier. That's true. Vitamin C is an anti-inflammatory agent. It's an antioxidant, for example, and it reduces throat and nose irritation. So there's some observations. Hypothesis: consuming vitamin C decreases the risk of catching a cold. So here we're using essentially inductive reasoning. We're combining specific observations to come up with a general principle. Now we don't know that this is true, right? It's just it's essentially an educated guess. But we come up with this positive statement saying, look, consuming vitamin C decreases the risk of catching a cold. That's potentially testable, and you could come up then with a prediction, right? If I consume vitamin C, I may have a lower risk of catching a cold. If I abstain, and however, or you could do it the other, you could even do it the other way. If someone abstains completely from vitamin C or any fresh fruit or vegetables, they will probably be sicker, right? They'll have a higher chance of catching a cold. Okay, so you can see how this would lead to experiments. No, I should say very briefly here that there's this crazy idea that taking mega doses of vitamin C is somehow a good idea. It's actually a terrible idea. That's essentially a completely, just having normal amount of vitamin C in your regular diet is okay, or the vitamin C you get from a multivitamin. You do not need mega doses of vitamin C. There's absolutely no scientific evidence that supports that that's at all healthy. Okay. So here's a hypothesis: consuming vitamin C decreases the risk of catching a cold. It's sensible, right? Needs to be tested, may be proven false, right? All right. So here's a question. What step in the scientific method involves making a tentative statement that explains an observation or answers a question? go back here. Okay, so we'll go ahead and start the question. I'll start the timer there. Go ahead and talk if you want. Talk to your neighbors, confer. See, as soon as I say talk to your neighbors, everyone's silent. It's weird how that works. All right. Get your answer in there. Remember, I'm only giving you 30 seconds here because we want to move on quickly. No? Okay, some people are hesitating. They don't want to respond. All right, that time's up. All right. Let's see your responses. Okay, here are our responses. Okay. What step in the scientific method involves making a tentative statement that explains an observation or answers a question? So what do you guys think? So you're pretty well spread out here. It turns out that C is going to be correct, right? That's the correct answer. So why is that the correct answer? Well, you're making a tentative statement, right? So that's the hypothesis, okay? The very first step is to observe, do inductive reasoning, which is a kind of generalization, right? The next step is the hypothesis. Once you've done the hypothesis, then you use deductive reasoning, which is an if-then statement. So that actually comes later, okay? All right. Very good, let's go on. Oh, I've got another question, sorry. More questions over. Okay, just one more. Okay, which statement is a scientific hypothesis? The time around. Okay. All right, put your answers in. Go ahead and look at the responses. So we've got, let's do this in order. We got rock music is better than classical music. No one picked that. That's kind of just subjective, right? So it depends on the person, right? Some people like classical music, some people like rock music. All right, next one is quite popular. Garden gnomes make strawberries grow larger. That's actually, you know, I kind of think that that's not terrible. But that's not correct. But what's that? Oh, you didn't mean to. Well, two other people agreed with you though. So, but here's some support for this one. This is testable, right? You could put a garden gnome out there and have two patches of strawberries, right? One with a garden gnome, one without, and see what happens, right? So it's perfectly testable. What's sort of against this is there's really no evidence, at least to begin with, to think the garden gnomes, which the little ceramic garden gnomes would actually have any effect on your. So yeah, probably not the best one here. Okay, answer here. Buying recycled paper is an acceptable practice. Of course it is, right? It's very good. But that has nothing to do with necessarily with science. It's a good thing to do. And then D, increasing dietary fiber decreases colon risk. That's actually the best one here, right? So that's the correct one here because there's actually some evidence for this, right? There have been studies already that have shown that, hey, look, probably eating whole fruits and whole vegetables is a really good thing because that contains fiber. People who eat these, very healthy things tend to be healthy. So that right there, maybe there's something to this. And then what you would have to do is actually do a controlled study to see if it actually does have anything to do with colon cancer risk or not. But there's some evidence, there's some basic evidence. Like if you have a lot of fiber in your diet, it means that things move sort of efficiently through the GI tract. That's probably good for getting rid of toxins and things like that that might be sticking around otherwise, right? So this is it's scientific because there's it's an educated guess that makes sense from a scientific point of view. And also it's testable, right? You could come up with a study with thousands of people and see if it really does make a difference. It's generally, by the way, considered to be good to have fiber in your diet. All right, so let's look at how this works. So we're going to go through this a little more stepwise. So we have the logical hypothesis test. Hypothesis testing is based on deductive reasoning. So we have a general principle to protect. The general principle is to predict observations. And so we're going to have if-then statements for making these predictions. So again, predictions are if-then statements. So let's start with this person here. We're doing the vitamin C example here. Here's our hypothesis. We should start with a hypothesis that's testable and is potentially falsifiable, meaning we could reject the hypothesis. Consuming vitamin C reduces the risk of catching a cold. Again, that's kind of a reasonable starting point because we know that people who eat fruits and vegetables are generally pretty healthy. We're going to make a prediction. If vitamin C decreases the risk of catching a cold, then people who take vitamin C supplements will experience fewer colds than people who do not. Okay, so that's reasonable. Test the prediction. Now we're doing an experiment, conducting an experiment or survey to compare the number of colds in people who do and do not take the vitamin C supplements. Okay. Okay, so here's a couple possible outcomes, right? If people who take vitamin C suffer fewer colds than those who do not, well, our prediction is kind of true, right? But that means that we do not reject the hypothesis, and what we would say is our data or our results support the hypothesis, but we don't say we prove the hypothesis, right? That's not scientific to say we prove the hypothesis to be true. What's the other possibility? People who take vitamin C suffer the same number of colds or more than those who do not. Then here we conclude the prediction is false. We actually very confidently reject the hypothesis. It didn't really help, right? So that we can say, it didn't work. The vitamin C supplements did not help. Okay, that we can say with much more confidence than we can say that in the other direction. Okay. All right, so hopefully that's clear. And again, as I said, the reality is that certainly a little bit of vitamin C is good. So a regular multivitamin or fruits and vegetables is the best way to get vitamin C. And taking megadoses is completely unscientific, and that's been shown to be completely, it seems sensible, but it doesn't make it's actually potentially harmful to take megadoses of vitamins. Okay, so how do we do an actual experiment? Okay, so this is something you've probably heard about a lot during COVID-19, or what people really want is a really good experiment. And what do you mean by that? Well, a controlled experiment. A controlled experiment is one where it's very carefully planned out and executed. You have a manipulation of the natural world that's going to be used to test your prediction. In a controlled experiment, there are typically minimum 2 designated groups. There could be more, but let's stick with 2 to keep things simple. We're going to have an experimental group that receives a treatment. We're going to have a control group that receives no treatment. So let me backtrack just for a second here, and I'm going to put up a graph here on the board. So we have two axes here, right? We have an x-axis and a y-axis. So what we're going to put here on the x-axis is what's called the independent variable. And on the y-axis, we're going to put something called the dependent variable. Okay. So this is the way most scientific graphs are done. An example of an independent variable might be the amount of fertilizer, right? So let's say we have different levels of fertilizer, just as an example. Fertilizer, right? So EG means example. So here's our example. We have different levels of fertilizer, 1, 2, 3. These are just arbitrary numbers. And then our dependent variables, we're a corn farmer out in Iowa. I used to live in Iowa. And the more, let's say the more fertilizer, the better. Actually, let's put another point up here, 4. Let's imagine that this is our data, right? So as we increase the amount of fertilizer, this is our corn yield, right? And we might be measuring the mass, right? How many tons of corn or something we're getting. As we increase the amount of fertilizer, it goes up, but eventually if you put too much on, you're actually starting to poison the fields, right? So you're starting to go down, right? So this is so on the x-axis, we have the independent variable. That's the thing that we're manipulating, right? You as a farmer in Iowa are changing the amount of fertilizer you're putting on your four different experimental plots. And then the amount of corn you get, and let's imagine the rainfall and sun is the same on all 4 fields, right? This is the variable that depends on the independent variable, right? This is the thing you're changing. This is the thing you're measuring, right? So that's the dependent variable. So these names actually make sense, right? This is the independent variable, always on the x-axis, dependent variable, always on the y-axis, okay? So they're using a different terminology here, which is sometimes the independent variable is called the experimental variable, right? That's perfectly fine. And the dependent variable is sometimes called the response variable, okay? Actually, independent variable, dependent variable are more the standard way of talking about these things. Okay. So again, the independent variable is the factor that's being manipulated or tested. And then the dependent variable is the response or change that occurs to the experimental variable. Okay. So we have 2 groups, experimental group and a control group. Minimum, you may have more groups. So data represents the results of an experiment. You would represent these in a variety of formats, such as tables or graphs. You'll use statistics if you're a doctor or a scientist. Standard error and standard deviations are both measures of uncertainty of a value. If you take a statistics class, you'll get a lot of information on how those are slightly different. Statistical significance determines whether results were due to chance. If the results are well supported by statistical analysis, a research paper is written and sent for publication in a journal. Okay, so that's basically how we would conduct, a controlled experiment. You have, if this were a clinical trial, instead of a corn in a field, you would have maybe different dosages, right, of a medication that might reduce blood pressure. And then on the y-axis, the dependent variable, you have blood pressure, right? So you can, it basically applies to any sort of clinical situation also very easily, right? All right, let's talk about a scientific theory. Scientific theory is much broader in scope than a hypothesis. A well-tested hypothesis may become a theory eventually. A theory is the highest status that any hypothesis can achieve. So let's talk about this word theory. So in ordinary everyday language, we use the term theory in a very loose manner. We say there were no cookies left in the cookie jar. There are 2 young kids in the family. I theorize that the kids, I mean, you probably wouldn't talk this way, but you could. I theorize that someone's been dipping into the cookie jar and stealing the cookie or getting the cookies when they're not supposed to, right? So that's the way you could use, that's a little ridiculous, but you could use the word theory in everyday ordinary language that way. But the term scientific theory has a much more weight than that. It's not really a hypothesis anymore. It's something that's been tested very thoroughly. There's a tremendous amount of evidence supporting it. One theory, there's things like Newton's law of gravitation, right? So that's one that's pretty well established. Newton came up with rules talking about how objects interact that have mass. And then just tremendous number of experiments supported him. Eventually, this person called Einstein came along, and he had an even better theory that explains things even better. And so again, people tested Einstein's theories with experiments, and they showed that he was even, he's even more correct in a sense. But Newton wasn't necessarily incorrect, but it's just when you push things to some limits, you don't. His theories don't give you exactly the right predictions anymore. Another theory that's of course very important for this class would be the theory of evolution. So evolution has essentially defined lots of things, like how your body works, the form of animals and plants, and so forth. So when we talk about the theory of evolution, it's no longer really just an idea that we're just throwing this out. Hey, I got a theory about this. No, it's been tested many times in many different ways using different techniques all across the globe by different scientists working independently. So it's much broader. A theory is much broader than a hypothesis. It's been extensively tested. It's supported over time. In other words, people started testing Darwin's ideas about the theory of evolution 100 years ago, and they're still testing it, right? They never stopped. A good theory has predictive value. So that's what's this next statement here. You can explain a broad range of facts. So something that's really useful is predictive, right? It can predict things about how the human body will respond to, a new environment. Like if you put humans on Mars, what would happen? Well, they might grow differently, that sort of thing, right? You could make all sorts of predictions based on evolution and so forth. And it should have a high degree of reliability. That's actually very similar to being talked about extensively testing. As we test this idea over and over again, we don't really see a lot of problems with it. Although scientists always argue about the details, right? There's huge conferences of people arguing about evolution, about how it really works. And that's not necessarily the weakness of science. It means that there's still, inquiring scientists trying to understand how it really works, and they're still trying to iron out the details. Okay, this is an interesting question. Let's do this one next. Okay, there isn't a question that science can't address. It's kind of a weirdly, it's like two negatives within one sentence, right? But basically it's saying there isn't a question that science can't address. That means if I turn around that and make it a positive statement, this means science can address all questions, is what they're saying, right? That's a simpler way of stating the same thing. So what do you think about this one? All right, I'm gonna go ahead and get your responses in. We'll take a look. All right, so we got false and true. Okay, so basically false is correct. Why is this correct? So you might think, well, science can address absolutely everything, but there's actually things that are sort of outside of the realm of science. One of course, would be things like the study of paranormal, events and things like that. Like if something's like a ghost that has no material, no matter or anything like that, doesn't interact, well, there's no way for scientists to explain something like that relies on explanations that go beyond science, right? And the other thing, do you have a question or no? Okay. I thought you were okay. The other thing, of course, so that's one big category that science doesn't really deal with. But the other big category really has to do with things like ethics and morals and things like that. Now, are actual scientists ethical and moral? Hopefully, right? And doctors too, right? And they do have conferences where they talk about the ethics and morals and things like that related to scientific type things. But in general, those sorts of things are really more at the level of the whole society, right? We have to deal with scientists and doctors and so forth can give information that might be helpful for an ethical or moral debate, right? But they're really it's a little more of a level of a whole society where we just debate these things. We
Would
do
some
have
of the questions also be like philosophical
yeah,
questions?
philosophical questions would fall in the same category, right? Although there is some interesting overlap, right? Like physicists and people that talk about philosophical debates or like the meaning of nothing and things like that often do get together and they do talk. And actually, so that there is communication, obviously, between greater society, like religion and religious people and scientists and so forth. And actually, these are often very productive. I used to work at Emory University many years ago, and we there was a big, that's actually a Methodist school, and they had a connection with Buddhists. So every summer there were a bunch of Buddhist monks on campus, and you see them in their wonderful, beautiful robes and so forth. And they were there to debate and talk with scientists and so forth and doctors. So there is communication, right? But there are questions that are sort of outside of the realm of science. That is what I'm saying. All right. So let's look at an example here. Design an experiment to test the drug effect. So most ulcers are caused by the bacterium Helicobacter pylori. Many years ago, someone was, I don't know if you're aware of this, but years ago, whenever someone would get an ulcer, the doctor would turn around and say, well, you're probably stressed. Look at your job, you're stressed. You should take a vacation. The person's like, well, I can't take a vacation. I have a family to support and all this stuff. But basically, the doctor would essentially tell you that it's your sort of psychological, not psychological, but you're stressed from your job or something like that. And they'd leave it at that, and then the person would go home being very disappointed and still have the ulcer and be very painful. Well, these two guys, Dr. Barry Marshall and Dr. Robert Warren, they had a different idea, which is that maybe that's not the cause of it. Maybe it's actually a bacterium that causes it. And they had so much trouble getting this point of view across that whenever they propose to do experiments, it gets obviously costs money to do experiments. They would not get funding and so forth, and it's just meet a lot of skepticism. So eventually what one of these two did was he essentially made a smoothie containing this bacterium Helicobacter pylori. He drank it, he gave himself ulcers, which of course are very painful, terrible. But he knew that he could cure himself by giving himself an antibiotic. So then a week later, he gave himself an antibiotic and he cured himself of ulcers. So he sort of proved through this whole, oh, it's just your job, he threw that out completely and just showed all these people that they were wrong. And of course, they went on and studied it very extensively. And now we know that Helicobacter pylori is in fact the cause of most ulcers, probably like 80% of them or so forth. Okay, so let's do this. New hypothesis, newly discovered antibiotic B is better treatment for ulcers than antibiotic A, which is in current use. Prediction, antibiotic, so that's our hypothesis, right? It's a simple statement, it's fairly general. A prediction gets us closer to doing experiment. It's going to be an if-then statement. If antibiotic B is a better treatment for ulcers, oh, well, okay, I have to fix this. We can fix this, right? If antibiotic B is a better treatment for ulcers than antibiotic A, then the patients given antibiotic B will show fewer symptoms, right? Okay, so I have to fix that a little bit. Turn that into an if-then statement. All right, so experimental design. We have a control group. We have test group 1 that receives the old antibiotic A. We have test group 2 receives antibiotic B. After 2 weeks of administration of the same medication or a placebo is essentially just a sugar pill. It's a pill that doesn't contain any medicine. In the same way, the stomach and the intestinal linings of each subject are examined to determine if ulcers are still present. Here they're showing the effectiveness of the treatment. So there was the control group that was given the placebo, nothing, so no antibiotic. Test group 1 gets the old antibiotic, so they show some improvement. They get essentially 60% improvement. In test group B with a new antibiotic, they show about 80%. So they're showing even more improvement, but there is variation, which is shown with these standard these error bars, right? So we stated the hypothesis, performed an experiment. We have our control groups. So here are the results. After 2 weeks, a higher percentage of those treated with antibiotic B were effectively treated. This result is statistically significant. Conclusion, based on the data that the investigators concluded, their hypothesis has been proven. Now, we can't say proven, right? But supported, right? Supported by the data. All right, so here's some review questions. So for each chapter, at the very end, you'll have a number of review questions. And just from experience, and I've talked to students, how did, I'll tell you that, so for your exams, you'll have about 80% multiple choice questions, right? So basically this will already help you with that portion of the test. And then there's another 20% that's gonna be things like answering diagrams, short answer, fill in the blanks, that sort of stuff, matching. Again, so answering all these questions will be helpful because often the teacher will look to these questions that we give you as review questions and say, hey, this would be a great short answer question, right? So this is a big hit, right? Take a look at this, these questions, see if you can answer them. And again, we're available for you. So if you have trouble, you can come to me. I'll give you tutoring information. I know we have wonderful tutors at Academic Services. I'll give you the exact. I haven't been given the times yet when the tutor is going to be there. As soon as I have that information, I'll get it to you. Again, so I post these every day. Like you have this already, right? It's if you go. Let me do a quick jump out of here. Let's go to Blackboard. So if we go into our course. Here. And you can see lecture materials, right? So I gave you the course introduction, you have the study of life, which is this first chapter right now that was more or less completed. Then the next chapter coming up is molecules of life. So you'll see this being populated with lecture materials. I have a reading guide here, so you can look at chapter 1. You can click on this. It's a Word file. It just gives. It's not that extensive, but it gives you a little bit of an outline of what what was in the chapter. Now, and again, we need to. I want to ask you. So, have you all been able to get in there and get in to see the textbook and everything? Okay. Who has not? Okay. If you haven't been able to, you don't have to raise your hand up. You can come talk to me. We got to get you in there. You go to My Connect section, right? It's not going to have any grades or anything here. Don't get worried here. But you would see a list of assignments, okay? So there's the Connect orientation. That's not going to count for any points, but it just gets you oriented. Then you have chapter 1, which is due basically, I believe this is Tuesday, next Tuesday, right? It's the day after Labor Day. And then we have the quiz. Now, normally I put like a little bit of a space of 2 days between the homework and the quiz, but I just moved the homework back because of Labor Day. Now, normally your homework assignments will always be due on Sundays, and then I believe the quizzes are always due on Tuesdays, okay? But that's a little unusual. So let's go back here to, Blackboard. So I want to talk a little bit about lab for a second. I believe, did you all do the lab safety sheet yet? If not, I can give you a safety sheet. Now, let's go to the lab for a second. So we'll go here. So 140L, right? That's our lab. So we did lab safety. If you didn't sign that yet, I think almost everyone signed it, but I have more handouts and stuff for that. Now we'll go to lab handouts. So next week we have microscopy. Now some of you I know are with me, some of you with a different lab, right? But this would have pertained to them. Go ahead, sure, question.
I don't know lab handouts for lab safety.
Right. Oh, you don't?
Yeah, I don't have those.
Oh, are you in a different? So I have lab section 1. Are you in different? Is your lab section 1?
Mine doesn't say it has a section on it.
Okay, it comes right after. You see how, look at where I'm pointing here?
Are you in the lab class?
By 140L, and then you see this /1.
I'm in 2.
You're in 2. Okay. You have 2. You know what, you can email me and I can send it to you directly. You know what I can do is I'll put the lab handouts, I'll duplicate it after class, I'll duplicate it under lecture also. So you have it a second place, okay? Okay, I'll do that, that'll solve that problem. But here's the thing, like,
Well, it is in here, except it doesn't have the like lab safety. It doesn't have its own tab.
right,
There's like, yeah, when it going to, it says in the learning modules, it says lab safety and conduct form,
right,
and then lab microscopy and all the other.
but, right, but do you have the actual lab though? Can you click on that and download it? Okay, so I'm bringing up the lab, so for next week, remember, it's gonna be the same pattern over and over again. Oh, geez. So you have, for next week, the pre-lab, which is due at the very beginning of lab, okay? Now you may have lab with me or someone else, it'll be the same pattern now. You have this thing to print out, right? You're gonna print that out. Have you all figured out how to use your printers yet? Okay, you all have a printer. You need to figure it out, okay? Because you're gonna have to start printing stuff out. You're gonna print this out. In this case, you don't have to type it here, right? You just write with a pencil or pen the names of all the parts of the microscope, right? And that's all there is to the pre-lab for next week, right? So it's fairly simple. Then you're gonna go through and do the lab. It's actually kind of a fun lab. We've done it many times where you look at cheek cells and things like that. And at the very end, oh, and you do some pictures, right? Do some drawing, which is fun. So then these are worth points. So then you'd be doing that and you're gonna fill this out. Now this should all be typed, okay? So what you could do, so the preference is that we want you guys to get used to the idea. These aren't like full like lab reports, but in a maybe a more advanced class, you might be typing out your lab reports and so forth. So we prefer, in fact, I'm gonna insist that you guys type everything in here, right? And then, so what you could do is come to lab with this printed out, right? Having this, or bring your computer so you can look at this, right? But actually you should print this out because actually this, you're gonna have to draw in here, right? So next week, you're gonna come with this, print it out. I would print the whole thing out probably. And then what you're gonna have to turn in are you don't have to turn everything, but you turn in for the post lab, you turn in your drawings. You would turn in this, and what you've typed in answers, right? So what you could do during lab is you could just write the answers in pencil or pen, and then type it up later. So it'll be all nice and neat. And then you turn that in as an actual physical document again, right? Because I can't deal with this. Sorry, this is a big class. So if you're all emailing me stuff, it's going to get very confusing very quickly. So you bring this as a typed up document prior to the next lab, the following week, right? Does that kind of make sense? I went through that before, but I think once we get into the rhythm of things, it'll be fairly easy, right? All right. So I am gonna stop there because we're gonna go into chapter 2, but we only have a few minutes. I'm gonna start that up next time. Okay. If you have any questions, I'll hang around and answer them. Okay. All right, thanks, guys. Yep. Go ahead.
That we're doing today.
Today is this week is just lab safety. There's nothing you have to do except
Just fill in the form.
yeah, there's like you have to sign the form and things like that, right?
Okay. I thought there was just the lab safety.
Yeah, this is a very, for most classes, this is kind of like a safe, not a safe, simple thing like lab safety.
Yeah, because I thought the lab safety was like a whole lab where it would just be like we actually go around looking and seeing what were the things.
Yeah, you're gonna point out, you have to know where the, what to do in case you get acid or something in your eyes. You have to know what to do in case there's a fire, all that sort of stuff, how to dispose of biological waste, use of gloves, things like that, all that stuff goes over. It takes about 20 to 25 minutes total to go through all that. And then have you guys sign, so it's usually a fairly quick lab, okay? Okay. Okay. Can you, is this a copy for me or?
It's for you,
Okay.
yeah,
I appreciate that. You're gonna need to still tell me. when
yeah,
you're-
yeah,
And you said you're gonna answer this around?
yeah,
All right, we're gonna have to work on that. That's a little bit later though, right?
yeah.
Okay, let's talk about that. There's 2 other lab sections. Do you know what those sections are? Okay. So, yeah, so we'll figure out like which lab section you could go to. And then I'll contact that. We'll go back and forth to email, right? And then I'll tell that instructor, you'll come in. Okay. All right, good. Thank you very much.
We have our schedule too.
Okay.
You just wrote our names on it, this is for all the
Oh,
same
you're all in the
thing.
same thing, that's fine. Volleyball, wonderful.
Also, can I get the form where I can sign? My lab teacher didn't so the signing, and my link wouldn't work to print it out from him.
Yeah. Yeah, so you would want to give this then to your lab instructor?
I think you.
Yep.
Yeah.